Hi, this is lecture 16 of ZOO3649 Evolutionary Genetics, and we're going to continue with our last lecture in the Darwin and Mendel series section. And this is lecture 16, and we will be talking about the instances where organisms do not have a Mendelian inheritance. Okay, in the last couple of lectures we talked about Mendel's laws and how he discovered a way of inheritance which has become the Mendelian way of inheritance. However, he was, as I said to you, he was very lucky to choose the pea plant because they followed those rules like most other organisms. However, there are certain situations where Mendel's rules are not followed, okay? Where there's a deviation from Mendel's rules of inheritance. And we're going to discuss those deviations today. In general, there are about 10 ways in which genetic systems can slightly deviate from the Mendelian way. And these include, well, the most important one, which is lethal alleles, and that happens when a mutation occurs in an essential gene, okay? And that gene has been knocked out. What happens? It stops working, it becomes lethal, and usually recessive, but not always. Then there's incomplete dominance, and incomplete dominance is where the heterozygote phenotype is intermediate between the two homozygote phenotypes. So if you have a red and a white, then under incomplete dominance, heterozygotes are not going to look red, okay, if red is the dominant allele. They're going to look intermediate, which is pink, okay? Then there is codominance, where heterozygotes shows the traits of both alleles, like a patchy cow, okay? Then there's sex-linked inheritance, where because the sex chromosomes are not the same size, females have an X and an X, and males have an X and a Y, for example, in mammals. And so when genes are found on those chromosomes, their inheritance is not strictly Mendelian. And then we have overdominance, where the heterozygotes have a higher fitness than either of the homozygote phenotypes. Then you have pleiotropy, when there is one gene that can control several different traits or several linked traits. Sometimes the traits can actually be quite different when a gene is acting pleiotropically. Okay, then we have epistasis. You would have gotten a problem from epistasis in your practical guide. I hope that you're all following and filling in your practical guides. That's very important for your practical mark. Then we have polygeny, and this is when there are several different genes that are controlling a single phenotype, like skin color, for example. There are many genes involved in the color of our skin, okay? And yet, what is the phenotype? It's one color, right? You have a color. But many genes are going to make that color, okay? So then you have environmental effects, when the environment actually affects the phenotype. Remember, the phenotype is what? Genotype plus environment, okay? So now we're going to talk about, in that point, we're going to talk about the environmental effects. And lastly, we have organelle DNA, and as you know, organelle DNA is only uniparentally inherited. You know this very much from 2544 last year. And so they obviously do not have a Mendelian diploid kind of inheritance, do they? That's kind of obvious. So let's go through each of these in order. And I've put lethal alleles at the front because I personally think that lethal alleles are one of the most important things in genetics. And that is because it's only many things we were able to find out in genetics only because a gene got mutated and stopped working. Okay, so a lot of our basic information about what genes do, how they work, etc., are obtained through lethal alleles, okay, or deleterious alleles, as we sometimes call them. Okay, so if we study these lethal alleles, you can actually tell a lot about what's going on in the genome. So what is an essential gene where this mutation occurs? Well, an essential gene is one that is absolutely necessary for survival. Okay, about a third, yeah, 1 out of 3 of all the genes. You remember we have about 25,000 genes. About a third of them are absolutely essential. That means if you have a mutation in one of those genes, it could be very, very bad for you. Okay, remember, a lethal allele is normally only lethal. That, in other words, the organism dies when you have 2 copies of the lethal gene, right? You have 2 mutated copies that are not working. Okay? If you have 1 copy and you have a heterozygote, usually, and you're a heterozygote, so that means you have 1 mutated copy which is not working and 1 good copy which is working. Usually the good copy can save you. You don't have to die. Okay? But we will get into lethal alleles. So it can cause the death of an organism, right? Because the gene is not working. The essential gene is not working. Okay, so these are usually recessive. They can be dominant, okay, but they're usually recessive. And as I just said, their phenotype is expressed in the homozygote configuration. In other words, you need to have 2 mutated copies that are not working in order to die from the condition. All right, so, and why? I've just explained it. Because a normal allele that is not mutated, right, it can produce enough dosage to make up for the one that is mutated, right? So if you're a heterozygote, you don't normally feel the effect of the lethal gene. But get this, guys, this is where inbreeding is this is why inbreeding is very bad. You know what inbreeding is? We're going to get to it at the end of the module in molecular ecology, but I just want to give you a sneak preview, okay? So inbreeding is when is the undesirable condition where close relatives breed with each other, like brother and sister or cousins. Now the problem with those breedings, okay, and the reason why most animals avoid those kinds of breedings, okay, and humans also avoid those kinds of breedings. We don't normally go for our sisters, do we? Right. So why? Because say these lethal alleles are very common, okay? They are all over the genome. But usually, why are we not dead? Because we are homozygotes, like this here. It says why? Because we have our normal allele, okay? And the lethal allele is hiding behind the normal allele, okay? The normal allele is masking the lethal allele. But many of us have these lethal alleles in our genomes. So if we come from the same grandparents, like for example, if I'm a brother and sister or cousins, the chance is that we will have the same lethal alleles, right? In other words, 1 good copy and 1 bad copy of the same gene at the same locus. So now when you mate with your cousin or your sister, it could be that one of your offspring gets the copy of the bad mutation from your cousin and the copy of the bad mutation from you, and it's lethal. You see, this is why close matings and inbreeding should be avoided at all costs. But we're going to get into that in one of the last lectures of this module. It's one of the reasons why lethal alleles are so common. Guys, you will be questioned about lethal alleles. Please make sure you have your thinking caps on. All right, so there are other kinds of lethal alleles. For example, alleles that are lethal only on condition. So it doesn't kill the organism every time, but it will kill the organism only under certain conditions. Okay? For example, we have temperature sensitive Drosophila lethal mutants, right? So for example, we've mutated, not me myself personally, but other scientists, they've mutated Drosophila fly larvae. Okay, they've mutated them, and so with the lethal gene. And so when they turn the temperature above 30 degrees while the little flies, the maggots, are growing up, they all die. They all die. All right? So, but funny enough, if they grow the maggots, the little flies, up at 22 degrees, all of them survive. So that means that lethal gene that they give them, the temperature sensitive lethal, it's not always lethal, right? It's lethal under certain conditions. In this case, when the temperature gets too high. What about semi-lethal alleles? These mutations, okay, kill some of the homozygotes in the population, but they don't kill all the homozygotes in the population, okay? There are some environmental factors and maybe other genes in other parts of the genome that are helping to prevent the lethal condition, preventing the death of the organism, okay? So in those cases, they're called semi-lethal. So why do we call lethal alleles a deviation from Mendelism? That's obvious, right? Because there's no homozygote recessive, right? You don't have a homozygote recessive. Remember, what is the Mendelian ratio when you do a monohybrid crossing heterozygotes with each other, right? You will get 3 of the dominant phenotype, right? So big P, big P, big P, small p, big P, small p, 3 of them. And then you will have 1 small p, right? That's going to be the recessive homozygote. That one is not going to exist when when small p is a lethal allele. All right? When the small p is a lethal allele and 2 small p's come together, that guy is dead. He's not there. So now you come and you're counting all the individuals in the population. No? You don't find that one homozygote recessive. He's not there, he's dead. So you don't ever count that one. So you will never get a ratio of 3 to 1, will you? How can you get a ratio of 3 to 1? The 1 is missing. You are never going to count it. So that is why we say that the lethal alleles are deviation from Mendelism. Because Mendelism, you're expecting 3 to 1, right? You don't get that. You just get the 3. Genotype ratio of Mendelism, you're getting 1 through 2 to 1, right? Whereas with the lethal allele, you only find the homozygote dominant and the 2 heterozygotes. The last homozygote recessive is not there. You don't see it. And I just want to, before we get into this example here, I just want to tell you in your practical guide, there are many problems where there are lethal alleles. And it's giving you the frequency of, say, certain genotypes, and you're struggling. You're thinking, "Aish, Marie, Prof has not given me all the information to answer this question." When you know what I'm talking about. Those who have looked at the practical guides already, you know what I'm talking about. But I have given you enough information. Just remember, in a situation where we are dealing with a lethal allele in your practical guide as well, when the allele is lethal, you are not going to count those ones, right? So you assume the frequency of that one is 0. Okay, the frequency of the lethal allele is 0. Or, sorry, the frequency of the genotype, small p, or the homozygous recessive, is 0. Okay, and if you know that, you can work out the rest of those problems. Okay, so let's look at this example of a lethal allele that is dominant. It's a dominant lethal allele, okay? So how does a dominant lethal allele now work? You see here, you have this, what you call the creeper gene, okay? A creeper gene in chickens, right? So if you have a creeper chicken, they look like this. You see where my mouse is here? Normally chickens have feet. These ones, you see, they don't have feet. Their feet are messed up, right? So they can only creep like this along the ground. But if you get a dominant creeper phenotype, so big C, the creeper gene is the C, big C. If you get a big C, you're dead. Okay? if you get a big C, small c, so if you're a heterozygote for the creeper gene, then you get those crazy feet that you see in the picture there with those chickens. Okay, so you have to be big C, small c, heterozygote to get that creeper phenotype. If you take a creeper individual, which is big C, small c, and you mate it with a normal individual, which is the recessive, right? So the normal, because creeper is a dominant allele, the normal individual would be small c, like this. So you mate small c with big C, you do a Punnett square, and what do you get? 50% of them have creeper, 50, so that they are looking like this, they are unable to walk properly, and 50% are normal. Right, so now what happens if we take a creeper versus creeper? So a big C, mated with a big C. Remember, creeper, when it is in a homozygous state, it is lethal. So what are you going to see when you mate creeper with creeper? So you're making big C, small c with big C, small c. So two heterozygotes are mating. Then what do you get? You get this typical, this is a Mendelian ratio. It is 1 to 1 to 2. Okay? One homozygote recessive, two heterozygotes, one homozygous dominant. That is a Mendelian ratio. Except what? Big C is lethal. It's dead. So you're coming to that population, you're counting all the chickens, and you're counting all the genotypes. You're not going to see any big C. So if you encounter a problem like that in your practical guide, assume the homozygote is 0. The homozygote condition is genotype frequency is 0 because it's not there. Okay? It's dead. And the same you get here, one of these, the big C is lethal. That combination of big C is lethal, and those chickens never get born. Okay? So what are you left with actually? You're left with the two homozygotes and the two heterozygotes and the homozygous recessive. Okay? In this particular situation. And the Mendelian ratio is what? 2 to 1. So 2 homozygotes to 1 recessive. We should have 1 to 2 to 1, shouldn't we? There's the 1 there. It's dead. So we don't count it. That is why lethal alleles are a deviation from 1 to 2 to 1 Mendelian ratios, genotype ratios, or phenotype ratios 3 to 1. All right. So incomplete dominance. Remember, we've been through all of this in second year, okay? Which is why I'm going through a bit faster in third year, because you know all this already. It's in your practical guide. It's everywhere. You need to know it. And you should already be remembering from second year. So incomplete dominance is, as I say, is almost like what Darwin said with blending inheritance, where when you have a parent that is one color, another parent who's another color, and the offspring is in the middle. It's got an in the middle color, an intermediate color. So, for example, if you take flowers, you can take flowers, but even if you take humans, imagine now you've seen what happens when there are mixed couples, right? Where you have a black and a white person married together and having kids. Like what color are the kids? What color are the kids? What color are the kids from when a black person and a white person get married and have kids? What color are those kids? Are they black? Are they white? No, they're not. They all look brown, right? They all look in between black and white. They don't look black, they don't look white. They look in between. Okay, so this is what we are calling incomplete dominance. All right? So where the heterozygote actually looks in between the two homozygotes. An example of this in plants is what we call this 4 o'clock plant. So look at this plant here, right? Look at this plant. This flower here has a genotype for a certain color. This flower here has a genotype for another color. If you mate those two together, you will get a flower of this color. Remember, this is not a Mendelian inheritance, right? Mendelian will say you should get, if you made these two together, you should get all of them looking that color there, right? Or if you made heterozygotes that all look like this, you should get 3 of these two, 1 of these. You should never get a pink, an intermediate. Nah? So, how does that work? Here it is. You have a red flower with a white flower, and they will give you a pink flower at the end. So these heterozygotes, where you have a dominant allele and a recessive allele, your heterozygote, that all heterozygotes have a pink color, as you see from this Punnett square here. 1 red, 1 white, and the 2 heterozygotes are pink. What is the Mendelian ratio? It's 1 to 2 to 1. Okay. But what would it normally be? Sorry. What would it normally be if there was no incomplete dominance? These 2 pinks, what color would they? They would be red, wouldn't they? Right? In a normal Mendelian inheritance. And so, what would the Mendelian ratio be? It would be 3 to 1. Right? Okay. Now we understand that. All right? So, that is what we call incomplete dominance. And why is incomplete dominance like that? Why? It's because what? The dosage, so this white allele here is not making any color. It's making no color. It's mutated. That's why it's white. Okay? It can't make a color. This one is making the color. But because the heterozygotes are pink and not red, what is it telling you about the dosage of that allele, the red allele, that one there? The dosage is not enough to produce the red color, right? It's only half the dosage, and therefore it produces the pink color. All right, so that's incomplete dominance. What is codominance? Now, it's when you have 2 alleles, but each of them code for a different color, and you see the different colors, right? And this is, for example, patchy dogs, patchy cows. You've seen the patchy skin of a cow, no? All of us have seen that. Beautiful, actually. But how genetically is it formed, that patchy skin? Now, look at this patchy skinned cow here. Yeah, you see where my mouse is? Right. This is codominance. It's because a pure red and a pure white cow mated, and they gave us this, what we call a roan cow. A cow with parts of it brown and parts of it white. Okay, so in the white parts, those alleles, that's the recessive allele together there, and in the cells where we have the brown part, that's the dominant allele, dominant genotype being expressed in those brown cells. Okay, so the and again, there are problems with codominance in your practical guide. Please don't delay in filling all those things out. Okay, so your homozygotes give particular colors, red and white, but the heterozygote is patchy. Nah? And that is what codominance is all about. And why? What Mendelian ratio do we have here? We have a 1 to 2 to 1 genotype ratio, as we would always have. But what happens, what is the phenotype ratio here? The phenotype ratio from this combination under normal Mendelian inheritance should be what? It should be 3 red to 1 white, as you know now. And if you are unsure why I am saying there should be 3 red to 1 white in normal Mendelian inheritance, go back to the very first lecture and start all over again until you get it. Okay? So, we should see 3 to 1, but we don't see 3 to 1. We see 1 red, 2 patchy, and 1 white. That is why this is codominance is a deviation from Mendelism. Okay, what about sex-linked inheritance? This is when the gene for a particular trait is located on the sex chromosomes. And here, if you're unsure, if you've never seen what a sex chromosome looks like, here they are. So on the left here, we've got our female sex chromosome. So the first one, and you see there are 2. One is given from mom, one is given from dad, right? Remember, dad can give an X because dad also has an X. Because here's a male, and all males have 1 X and 1 Y. You see this Y, it's half the size, it's even less than half the size of that X. So for example, If there's a gene, right, and this is a male here, here's a female. If there's a gene and the gene is here, where you see where I put my mouse here, the gene is there, right there, on that part of the chromosome. So, in other words, that part of the chromosome, there. This female here will have a copy of the gene there and a copy of the gene there. You see that? Marlan, let's look at the male. The gene is on the chromosome, on this part of the chromosome. The male will have one copy of it there. Mar, what about this part? This part here, where the gene should be, is missing. That's why we call it sex linked. It's missing. So this male, if there's a gene here, he will only have one allele. He will not have two like a normal diploid should, right? He will only have one allele. In other words, he will only get it like a uniparental inheritance, similar to chloroplast and mitochondrial DNA, except this is Y chromosome, and you can get also a uniparental inheritance, not a Mendelian inheritance, because why? This part of the chromosome is missing. So if there's a gene here, whatever that gene is, the male will see the phenotype. Because why? There's not another piece here. If there's a lethal gene here, this male will die. Because why? There's not another one here, a normal one to help the lethal. Remember, the lethal is only lethal when it's in a homozygous, right? But if you have a situation here like this, where the male cannot be homozygous, he only has one copy, the other one is just not there. That Y chromosome is not, that piece is not there. So if you have a copy here, you are going to be, immediately, it's going to be your only copy. And you will have that phenotype. All right. So in females, the inheritance is Mendelian because they have two normal chromosomes. But in males, one copy of the recessive allele will show the phenotype. So if you have any copy here, if you have any allele, whether it's dominant or recessive, that's the phenotype you will show. All right. And if you've got a recessive allele, then you're in big trouble, because you have got no other allele to mask the dosage. Okay, so let's look at an example. Okay, now here is exactly what I was saying to you. Here is the X and the Y of this man, right? Here is the X and the X of this woman. Now these two are married and they have kids. They have these 4 kids here. Now, so look at the inheritance here. Now this male, there's the gene we're talking about. It's the gene has to do with color blindness. Okay, and again, you have many problems like this in your practical guide. You better be filling them out and doing those problems, because when you come for the practical, if you're for the face-to-face practicals, you better have filled out the previous practicals before you arrive in the lab. Okay, so look at this guy. Here's the gene. Here's the locus that we're interested in, this blue one here. Here it is. The white allele, the blue allele is what? It's a normal, fine, wild-type allele. There's nothing wrong with that allele, right? But the white allele is the mutated allele. This is the one that is not working, okay? So look at this man here. He has got one copy of the allele. He doesn't have another copy. But he's lucky, isn't he? Because his copy is a good one. So he is unaffected. This one is fine. Okay? This female is also fine. Why? Because she has got one good copy and she's got one bad copy. But at least she has one good copy, so she's also fine. The dosage of that copy is enough to make her fine. But here's the problem, right? The problem comes with the kids. The problem comes with the kids. So what happens now when we mate these two people? When we mate these two people, if you have a girl, okay? If you have a girl, the girl will be fine. She will be fine because she can get maybe 2 good alleles, right? The allele from dad and one of these good alleles from mom. She could also get, and she's always going to get the good allele from dad. So she's going to get that because she's a girl. She must be XX. So she's going to get that one from dad, that good one. But she could also get this bad one from mom. Okay? And in that situation, she's going to be just like mom. She's going to be a carrier of that lethal allele. All right? But look, she's okay. She doesn't feel any problems. Mom also doesn't feel any problems because the good allele, the dosage is enough. Now, so, and the girl kids also don't get problems. Where do the problems come? The problems come with the boy kids. Okay? So here we go. The boy kids are always the one who's affected. Now, the boy must always get a Y, right? That's what makes them a boy. And it must always get the Y from dad. So dad can only give the boy this one here. He can't give that one. If he gave that one, then it would be a girl. No? Dad must give only this one to a boy so that that will be a boy. Dad can't give his X because boys need an X and a Y. And mom doesn't have a Y to give. No? So if you're going to have a boy, the Y is coming from dad. Right? So let's look at the boys. This Y has come from dad. This boy was lucky because his X come from mom, but he got the good X. Right? Look, he got the good one. This boy, this boy is unlucky. Okay, this boy is very unlucky. So he has got obviously he's got the Y from dad because he's a boy, but he got this bad copy here from mom. So you see, he's got one bad copy, and on the other side, he's got no other copy, like this girl, for example. She's got a bad copy, but on the other side, she's got a good copy from mom, and she can, from dad, sorry, and she can mask, the dosage is enough that she doesn't feel affected by this condition. But this boy, he has no other copy except this bad one that the mom gave him. What dad gave him is the Y. The Y doesn't have a gene for that locus. Okay, so that poor boy is going to be very badly affected. All right, so the Mendelian ratio is 1 to 1. Okay, the Mendelian ratio is 1 to 1 because we don't see this lethal son here. Okay, so the Mendelian ratio is 1 to 1. Why do I say that? Why do I say it's 1 to 1? You're saying, Aish Marie, why do you come with 1 to 1? Well, I come with 1 to 1 because when you are a boy, the inheritance is 1 to 1 from your dad. Okay? The inheritance for boys is 1 to 1. Just like with mitochondrial DNA for girls. It's 1 to 1 with mom. They get the mitochondrial DNA only from mom. Okay? And it's the same here. With this, for males, with sex-linked inheritance for males, the ratio is always 1 to 1. He will get just the same as what dad got. Okay, dad passes it on in a 1 to 1 fashion. So now what's overdominance? Overdominance is where the heterozygote has a higher fitness. This is called heterosis. This can be called heterozygote advantage. And a common, and this is a beautiful African example, a common example is called sickle cell anemia, right? We've been through this again in 2544, but we'll go through it quickly here in 3649. This is an autosomal recessive disorder, and it affects individuals by producing abnormal hemoglobin. Now, which one here is abnormal? Obviously, this one is the normal one, this donut-shaped one, and this is the abnormal one, right? This donut-shaped one gives a big surface area for what? To attach oxygen, right? Because this is a hemoglobin cell. It's a red blood cell. When you cut your hand and red blood comes out, those are the things that are coming out. Millions of them, right? So each of them needs a surface area to attract oxygen. Because why? The blood has to come to the lungs, pick up the oxygen, and then go to the rest of the body and feed oxygen to all the other cells. So those things need to carry oxygen. If they are looking like this, they can't carry oxygen very well. Because look, the surface area of that one on the right is much smaller than the one on the left, the cell on the left. So, and on top of that, This one is round, okay? When you put round things together and you put them in a small place and you shake them, they generally can move through a tube or a pipe or something because they are round. Now, if you put this one here on the right, the sickle shape, huh? If you put them into a small place all together, what's going to happen? They're not round. They're going to get caught in each, tangled in each other. They're going to choke it up. They're going to get stuck, right? So this cell here has no advantage when you have when you're homozygote for you have two alleles giving you that on the right here, the sickle cell. You're dead. Okay, you can't live. It's a lethal condition. But if you're homozygote, how does it give you an advantage? You know, right? You know. So let's have a look. I've just explained it to you. I've just explained it to you. The sickle cells are not good, right? So if you have two sickle cells, you have a much shorter lifespan than unaffected, okay? And the Mendelian ratio is 1 to 2. But, because why? Why? Because it's lethal, right? It's the same as lethal alleles. But we have an added situation of overdominance here, right? Not just a lethal allele, but we have an overdominance because we know that this sickle cell, even though it's lethal if you have two homozygote recessives, two sickle cell alleles, but nevertheless we see a very high frequency. We find this sickle cell very high frequency in Africa. Look at here, in Congo area and in Nigeria, Cameroon area. Why? And also here in this part of Arabia, Dubai, Oman. So why do we find it? That is where malaria is found. Malaria is caused by the Plasmodium protozoan. How? What does this parasite do? What does this parasite do? Let me show you what this parasite does. Okay? This parasite comes into this cell. Okay? It finds this small hole here. And so it burrows into that. It actually starts living inside that little hole there. And you see it's protected all around. So when your immune system is trying to fight this, it can't find it because it's hiding inside that hole. Okay? And it's protected from all sides. But if you have the sickle cell here, where is the malaria going to hide? There's no hole for it to hide. It can't go, it can try to hide, but it's going to be attacked from this side. You see where my mouse is? So it can't hide properly in that sickle cell. So if you have the sickle cell, one sickle cell and one normal cell, you don't get malaria. Because the malaria finds it hard to hide in your cells. Even though you have one normal cell, it still finds it hard with those sickle cells around to hide. So that is why the sickle cell, even though it is lethal, it is found in high frequency where you have malaria. Because the parasite is going to go into the blood cell and mess that blood cell up. Okay? So if you're a heterozygote, you have a normal allele and one sickle allele. Okay? You actually have a higher fitness compared to homozygous, right? And you don't get malaria. Talking about fitness on different genotypes. What is the fitness of the heterozygote? What is the fitness of the homozygotes, etc.? Practicals 6, 7, and 8, we are going to be doing this. We are going to be measuring fitness on homozygotes and measuring the forces of evolution. So that is good. And then what? You're going to write your assignment 2 based on practicals 6, 7, and 8. So please don't not bother coming for those practicals. If you don't come, you will get 0 for your practical mark. And on top of that, what will happen to you? You will get a problem when you are doing assignment 2 because you won't know what to do. All right. So please make sure you're filling in those practical guides and preparing for the face-to-face pracs that we're going to have, and they're going to come soon. All right. So the ability of a gene to affect the organism in many ways. So one gene affecting many ways is called a pleiotropy. Okay, and again we can have the hemoglobin gene as a perfect example. Okay, so what happens? What happens when you have sickle cells? Okay, remember, in areas of high malaria, your fitness is high. Okay, but if you are not in an area of high malaria, then your fitness is not as high as the heterozygote. Why? Because the sickle cells, even though it's one gene that is being mutated, look at how many different things are caused, how many phenotypes are caused. One gene is mutated. That's what pleiotropy is. One gene affects in many ways, right? So one gene, the sickle cell gene, is mutated. So one mutation, it affects the breakdown of red blood cells. So it affects a phenotype like that. But it also makes them clump up and start clogging, right? As I explained to you a little earlier. So it has another phenotypic effect. And then it can also accumulate these sickle cells in an organ called the spleen. The spleen is the one that's responsible for making your blood, right? So it can accumulate because they get stuck in there, right? So these are three different phenotypes, all caused by the mutation of one gene. You see how one gene can affect many different genes. What about epistasis? Epistasis is really cool. You have at least one example in your practical guide of epistasis problem, and you're going to have to figure out what is going on with that epistatic interaction. When genes interact epistatically, it means that a gene in one part of the genome can alter or influence how a gene in another part of the genome, in maybe even another chromosome, is expressed. Okay? So the genes are affecting each other even though they are on different parts of the genome. That's interesting, right? Let's look at an example of that. Let's look at this black locus, okay? Let's look at this black locus. And black, we have big B or big B small b, right? We have black, right? So anything with a big B or a big B small b will be a black. Okay. small b is brown, okay? So that's at the B locus, okay? So now let's look at the C locus, which is another gene somewhere else in the genome, right? The C is also responsible for color, but not the black color. It is responsible for the white color, okay? So if you have a dominant allele, okay, then you have the expression of a color. All right? So basically, if you have big C or big C, you have a color. But what happens if you have small c? You have no color at all. Okay? So small c controls the white, and so C locus controls the white, and B locus controls the black. So this now leads us to what Mendelian ratio here? Normally we would get what? A 9 to 3 to 3, and this one, the homozygous recessive for both loci, that will be the only one, right? 9 to 3 to 3 to 1. But under epistasis, you don't get that. Under epistasis, you get the Mendelian ratio 9 to 3 to 4. Because the 1, this one here, is now added to the 3 because all 4 of these guys have no big C allele. You see, you must have a big C allele to have a color. Okay? If you don't have a big C allele, you are white. So that's why these 4 rats do not have a big C color, and therefore they are born white. Even though the Mendelian ratio says that 3 of them should have a color. Right? So there you go. That is why we call it a deviation from Mendelism. So quickly, polygeny. This is when many genes influence the same trait, and there is an additive effect from two or more genes. I published a paper about this in 2019, additive traits and hybrid speciation. Maybe if you're interested, you can go have a look at that. One of the best examples of a gene, of many genes contributing, is the skin color gene. In fact, you see here that there are two genes in these rats that are contributing to the color, right? But in reality, there are many more genes, including those two, that affect the color. In humans, in all kinds of animals, all right? And so pigmentation in humans is controlled by at least three genes, which are on different parts of the genome, different chromosomes, and they're interacting with each other. Their gene products are interacting with each other. So, okay, so our expected Mendelian ratio, if we had to do a trihybrid cross, remember, don't get confused, monohybrid crossed expected ratio is 3:1, okay, phenotype ratio. The dihybrid cross, so then two traits or two loci, are what? 9:3:1, right? Because you got 16 in total. What happens if you have 3 loci? I've not given you any problems like with 3 loci because they're very complicated. But the ratio will be, if you did a trihybrid cross and you worked out the Punnett square, it will be 27 to 9 to 9 to 9 to 3 to 3 to 3 to 1. That would be the Mendelian ratio, right? But for these genes here, we don't find this Mendelian ratio. We find this 1 to 6 to 16 to 20 to 16 to 6 to 1, where between 16, 20, and 16 here, in the middle of the graph, is the most common phenotypes that we find. Okay, so this is the Mendelian expectation. This is the actual reality. So that is why we call it a deviation from Mendelism. Genes in the environment, as you know, the phenotype is the product of the environment and the genotype. And so if the environment is doing something crazy, it could affect what phenotype you get, right? And in that case, you won't see a strict Mendelian ratio. So in this flower, like the snapdragon flower, you have the red, big R, mated with the recessive, small r, and what do you get? You get, if you grow of course you get heterozygotes, right? All the kids are heterozygotes from that mating. But if the heterozygote is grown in a bright light and cool temperature, all the heterozygotes will be red. Remember, you should see a 3:1 ratio. You're not seeing it. All of them will be red. You'll be like 4 out of 4 are red. But if you do the same mating and you grow the heterozygote kids in shady conditions under warm temperature, all of them will be white. Remember, only 1 out of 4 should be white in Mendelian. Here, all 4 are going to be white. So your ratio is messed up. Okay? Your ratio is messed up. Organelle DNA. So this is the last one. This is a very long lecture. I apologize, but I only apologize because of the downloading. Yeah, I don't apologize because you need to know all this information in order to pass the module. So organelle DNA, as you know, this is, I don't really have to explain this too much. Mitochondrial DNA, chloroplast DNA, you only get it from one parent. So immediately you're violating Mendel's law. Mendel's laws are for diploids. They are about segregation, about dominance and recessiveness. 2 alleles, how do the parental alleles interact, right? But when you have a uniparental inheritance like organelle DNA or Y chromosome even, what happens? You don't get any interaction because you only get one copy from One parent. You don't get another copy from another parent, and there's no chance for the copies to interact. Okay? There's no Mendelian anything. You just get one copy. And that's why it is a violation from Mendel's law. Look at this. Look at this. Look at this. Say these two people get married and have kids. They have these 3 kids, right? Mom has a disease that is in the mitochondrial DNA. Okay? Who's going to have the disease? All the kids are going to have the disease. Because why? All the kids get the mitochondrial DNA from mom. Dad gives no mitochondrial DNA whatsoever. What about this situation? When you have now mom is not affected, but dad is affected by a condition, right? On the mitochondrial DNA. What are the kids going to look like? Kids are going to be fine because the kids didn't get any mitochondrial DNA from dad. They only got it from mom. So that means if there's a mutation in the genes of the mitochondrial DNA, all the kids will get it, but only the daughters will be able to pass it to their kids. The boys will not pass it to their kids. Okay, and that ends our section on Mendelian inheritance. And the end of our Darwin and Mendel section. So stay tuned for the start of population genetics.